Hello everyone. Today we are going to continue with the topic nutrition. We were doing about the autotrophic nutrition in a previous video, and I told you that autotrophic nutrition, it takes place in the plants and it takes place in 4 conditions. That is, during the process of photosynthesis, plants need water, chlorophyll, sunlight and carbon dioxide. And even if one is missing out of these, photosynthesis will not take place. So today we are going to do a few activities, a few experiments to prove this, that all 4 conditions are important for the process of photosynthesis. The first activity we'll do to show that chlorophyll is necessary for the process of photosynthesis. In this activity, what we do is, we take a potted plant which has variegated leaves. Now these variegated leaves, what does that mean? Variegated, what does this word mean? This means the leaf which has other color also on it, other than green color, right? Like you all must have seen money plant or the crotons, right? Money plant, the leaf has some yellow patches on it. In crotons you can see light pink color or the dark maroonish color also sometimes, right? So these are known as variegated leaves, having patches of other color also on them other than green color, right? So here, as I have drawn this leaf having some red patches on it. So we take a potted plant which has got variegated leaves and keep it in dark room for 3 days. Why we do it like this? Why we put it in the dark room? As I told you previously, that the plants, they store their extra glucose which they prepare during the process of photosynthesis in the form of starch. So when we keep it in dark room, photosynthesis will not take place because there is no light, right? So it will use up all the starch which it has got in the stored form. So that it will use up in these 3 days, that will get over. Then we take that potted plant out and keep it in sunlight for 6 to 8 hours. Right? In these 6 to 8 hours, it will again prepare starch. Right? Again the process of photosynthesis will take place and it will prepare starch. Now what we do is, we pluck the leaf of this plant and on that plucked leaf we put a tracing paper. The tracing paper that you use for your drawing also. Right? When you have to trace something, you use a tracing paper. So same way here, we will put a tracing paper on this. Now suppose this is a tracing paper, you put it on the leaf and you draw the leaf. And wherever there are patches, you put them like this. Right? You sketch them. So on the tracing paper now you know on which places the other color spots are present. Now this leaf we put in the boiling water. In the boiling water, all the cells will be deactivated, they will be killed. That is why we put it in the boiling water. After that, we take a beaker, put some alcohol in it, and we put that alcohol on the water bath, and inside the alcohol we put the leaf. Now why we put this alcohol on water bath? Now what is a water bath? First of all, let me tell you. This water bath, we take, it is just like if we see it in the chemistry labs, this water bath, बिल्कुल जैसे लोहे की कढ़ाई होती है छोटी सी, ऐसा होता है वाटर बाथ, राइट। उसमें पानी डालकर उसमें हम ये बीकर डाल देते हैं जिसमें अल्कोहल है, and we put the leaf inside it. ऐसा क्यों करते हैं? जब हम इसको हीट करेंगे तो हीट जो है वो डायरेक्ट अल्कोहल को नहीं लगेगी। अगर हम इसको डायरेक्ट हीट पर रखते हैं, the alcohol will catch fire immediately. That is why we never boil alcohol directly on the flame. We put that beaker in the water bath. That is just kind of double boiler, right? And we heat it. Now when this water will start boiling in the water bath, naturally the alcohol will also get heated up and that also will start boiling. So what will happen? It will decolorize the leaf. All the chlorophyll which is present in the leaf will come in the alcohol. The alcohol will turn green in color and the leaf will become pale. Right? This leaf will become pale. Now we cannot see any patches on it. It is all of cream color. बिल्कुल plain cream color की leaf हो जाएगी. All the chlorophyll will be removed from it. Now what we do it? We rinse it in water and then we take a dilute solution of iodine. Iodine here we will use to test the presence of starch in the leaf. This iodine, it is yellowish brown in color. Now when we will put this iodine on this leaf, we will see that the portion which was of different color, that was not green in color, either it was having yellow color patch or red color patch on it, those patches will not show the blue-black color, and the portion which was green in color, that becomes blue-black in color. How will we come to know it? Come to know where the patches were? We will now put this tracing paper on this leaf, and we will see that wherever the patches were marked, they are of yellowish brown color, that is the color of the iodine, and rest of the portion which was green in color, that has become blue-black in color. This is the test for starch, for the presence of starch. Wherever the starch was present, that has become blue-black in color. So this shows that only the green portion of the leaf which was having chlorophyll, that prepared starch when it was put in the sunlight. So this proves that chlorophyll is very much important, very much necessary for the process of photosynthesis. Wherever chlorophyll was not present, there were different color patches, it did not prepare any starch. The second activity here we have taken to prove that carbon dioxide is necessary for the process of photosynthesis. For this also we take two potted plants. Here we are not taking any variegated leaves, simple two healthy potted plants. And these potted plants also we keep in the dark room for 3 days. Same way we de-starch it first of all. Then we take them out and we place these potted plants on the glass plates. Right? These are the glass plates. And near one of the potted plant, we will put a plate or a ball which is having KOH, that is potassium hydroxide in it. Right? And then we will put a glass bell jar over it. We cover it with a bell jar. Both. Now in one we have put the plate having KOH and in the other we have not put anything. Now we seal it right? We seal it, we make it airtight, we apply some Vaseline over here so that the bell jar is fixed on the glass plate. Now, why we have put KOH here, the main function of KOH here is it will absorb all the carbon dioxide which is present inside this glass bell jar. Right? When we are putting the bell jar, some air is also trapped in it and air has got carbon dioxide in it. So this KOH, it will absorb all the carbon dioxide. There will be no carbon dioxide left. Now when we will put it in sunlight for a few hours, maybe 2 or 3 hours we will put it, what we will do after that, we remove this bell jar and we pluck the leaf of both the plants. Now again we will start with the same process. First we will dip these leaves in different into separate apparatus. We will perform the experiments. We will put both first in the boiling water, then we put both in different beakers containing alcohol and we will boil them so that all the color is removed. After that we rinse them in water and then we test them by putting dilute solution of iodine. Right? Now what will we observe? We will observe that this leaf, the plant in which KOH was placed here, the leaf of that plant will not show any test for iodine, while the leaf of this plant will show a positive test for starch. It will turn blue-black in color. Why? Because here there was no carbon dioxide present. So photosynthesis did not take place when we had put it out in the sunlight. While here carbon dioxide was present here inside the bell jar. So what happened? The photosynthesis process took place and starch was produced. So this experiment proves that carbon dioxide is necessary for the process of photosynthesis. Now the rest two experiments to show sunlight and water, same way we can perform. For sunlight, like if we take a leaf of a healthy plant, same way first we put it, so the procedure will be same. First we put it in the dark room for 3 days so that all the starch is used up. Then we put it out in sunlight for 6 to 8 hours. Now when we have put it in sunlight, what we do is on one of the leaf, we put a black paper strip. Right? Suppose this is the potted plant and one of the leaf we put a black paper strip on it. Right? Suppose this is the plant and these are the leaves on it. Right? And on one of the leaf we put a black paper strip. We clip it from both, simply with the paper pens we clip them. And we put it out for 6 to 8 hours. Now after 6 to 8 hours, we pluck this leaf. Again the same procedure. First we'll put it in boiling water, then in alcohol or water bath. What will happen? It will get decolorized. Then we will put the iodine solution on it. So what will we observe? We will observe that this portion, which was covered with black paper strip, this will not show any test for starch. Rest of the portion, this will become blue-black in color. Why? Because here black paper strip was put. So the sunlight could not reach here and photosynthesis did not take place here in this part of the leaf. Right? Same way we can do it for water also. So this is how we show that all the 4 components, carbon dioxide, sunlight, water, everything, all 4, chlorophyll, they are important. Even if one is missing, photosynthesis will not take place. So carbon dioxide the plants get from the atmosphere, right? Water it absorbs through roots. Sunlight, of course, from the sun, and chlorophyll is present inside the chloroplast in the leaves. This is how the plants can prepare its food. Along with this, it also needs some other minerals, some other components, nitrogen, phosphorus, magnesium, iron, which it takes from the soil. Nitrogen is very, very important for plants, because nitrogen helps in making the proteins. But plants cannot take nitrogen directly. It always takes nitrogen in the form of nitrites or nitrates. Plants always take nitrogen in this form. Right? You have already done how nitrogen cycle. During nitrogen cycle, the nitrogen gets converted into nitrites or nitrates. So, this is all about autotrophic nutrition. And in my next topic now, I'll start with the heterotrophic nutrition, that is done by the animals, right? Because they depend on others for their food. They cannot prepare their own food. So I hope you have understood this much portion. Revise it, learn it, and next video then we'll start with the heterotrophic nutrition. Don't forget to like, share, comment and subscribe my video. Thank you.